Hi everyone, this is the YouTube channel of Chamel Dev Institute of Pharmacy. Today we will discuss about different types of microscopes used in microbiology. In this topic we will discuss about the introduction of microscope, basic principle of light microscopy, instrumentation and type and its application in microbiology. So we will discuss about the microscopy is defined as the use of a microscope to magnify and study the small objects that are too small to be visualized with the naked eye. Basically three main types: optical or light microscopy, first scanning probe microscopy, and last one is electron microscopy. So these are the three basic class: optical, scanning and electron microscopy. If we will discuss about some terminology, naked eye, we can see 0.1 mm structure. With the help of light microscope, we can see 0.1 micrometer, and with the electron microscopy, we can see 2.5 nanometer object with very easily. So in this, what we have to do? A light passes through a specimen through a single or a series of magnifying lenses to allow a magnified view of the sample. Important factors in light microscope include magnification, resolution and contrast. If we will discuss about what is microscope. So a microscope is an array of two lenses. Lenses include ocular lens and objective lens. Lens combine to enlarge the object. Next terminology is magnification. Total magnification achieved is the product of the magnification power of the lenses used. So microscope equal to objective lens into ocular lens. A simple microscope have a magnification power of 1000 to 1500, whereas electron microscope have magnification power greater than 2,50,000. If we will discuss about resolution, the resolving power is the ability of a lens to separate or distinguish small objects that are close together. Depends on the quality of lens and the wavelength of illuminating light. Shorter the wavelength, greater the resolution. Next one is contrast. Reflect the number of visible shades in the specimen is needed to make object stand out from the background, achieved through various staining techniques. Microorganisms are essentially transparent and must be stained for bright field microscopy. So right now we are coming on types of microscope. So basically we have in syllabus only in light microscope, but we will discuss about little bit classification of electron microscope. So first, electron microscope contain or light microscope have various type of microscope like compound microscope, bright field microscope, dark field microscope, phase contrast microscope and fluorescence microscope. While electron microscope contain transmission electron microscopes, we can say TEM, and scanning electron microscope, we can say SEM. So right now we are coming on compound microscope. Basically a compound microscope used in every lab. So this is the picture of compound microscope. It contains ocular lens, immersion oil, stage, condenser lens, light source, oil immersion objective lens, specimen on slide. So these are the component of this compound microscope. If we will come on next image. So firstly the light passes from downside and it goes through condenser, then object on a specimen, then objective lenses collect this eyes and transfer to the object lens, and we can see by the eyes very easily. The light microscopes use visible light or ultraviolet rays to illuminate a specimen. Two lenses in the compound microscope is ocular lens 10x, objective lens 4x, 10x, 20x, 40x and 100x. Resolution and contrast are controlled by condenser and iris. The condenser function to the focus light source on the specimen and provides an uniform illumination. An iris diaphragm controls the amount of light reaching the specimen. So this is the structure of compound microscope. Right now we are coming on use of compound microscope. So a compound microscope is used for viewing a small specimen, used to visualize different sample at high magnification 40 to 1000x, used to observe both prokaryotic and eukaryotic cells, to study cells and cell culture, plays vital role in the clinical laboratory, to detect the microorganism directly in clinical specimen, and to characterize the growth of microorganism in culture. Next category is dark field microscope. It is produce a bright image against the dark background. Dark field microscopy removes the unscattered beam from the image. Only light that has been reflected or refracted by the specimen forms the image. This is accomplished through the use of an annular aperture that will produce hollow cone of light that does not enter through the object lens. We will discuss about the application of dark field microscope. It is used to observe living unstained cells, used for visualizing different type of algae, used for viewing blood cells and bacteria. So this is dark field microscope. Next category is bright field microscope. Bright field microscope is the conventional technique produce a dark image against a brighter background. Bright field microscope used light from the lamp source to illuminate the specimen. This light is gathered into the condenser, then shaped into a cone where the apex is focused on the plane of the specimen. We will discuss about the application of bright field used for observing stained or naturally pigmented or highly contrast specimen, generally used with the compound microscope, used for visualizing different types of bacteria and cell structure. So this is all about bright field microscope. So this is the image of dark field and bright field microscopy, or we can say see the comparison. If we will talk about the bright field, there is a background is bright and the object is dark. In the dark field, there is a background is dark and the object is bright. Next one is phase contrast microscope. It is light is also oscillation and the phase changes when passing through an object between the light that pass through diffracted light and the remaining light, direct light. This phase contrast is converted into brightness differences to the observed specimen. A phase plate is located at the focal point of the light between the objective lens and the image surface so that only the phase of direct light changes. This generates contrast on the image surface. In phase contrast microscope, the annular ring in the objective lens and the condenser lamp is separated light. What is the use of phase contrast microscope? This is used to study prokaryotes and eukaryotic cells, used to observe living cells like bacterial components such as endospores and inclusion bodies. Transparent cells can be observed without staining them because it is not necessary to stain cells. Cell division and other processes can be observed in a living state. Next one or last one is fluorescence microscope. Many substances observing light from particular wavelength and energy emit light of a longer wavelength and lesser energy. Application of this phenomenon is based on fluorescence microscope. A specimen is exposed to the high energy short wavelength like UV, violet or blue light to excite the electron within the certain molecule inside a specimen. When the excited electron returns to their original energy level, they emit light of a lower energy wavelength in the visible spectrum, which forms the image of the specimen. So this is fluorescence microscope. Right now we are in continuous with fluorescence microscope. Usually specimen are stained with the fluorochrome or fluorophore. That fluorescence brightly upon exposure to the light of a specific wavelength. Barrier filter removes the remaining blue light and shows a bright image of the object resulting from the fluorescent light emitted by the specimen. What is the use of fluorescence microscope? It is basically used for imaging a structural component of cell, conducting viability study on cell population, imaging that genetic material within a cell like DNA and RNA, viewing specific cells within a larger population with techniques such as FISH. So this is all about microscope used in microbiology. If you want to more video, please subscribe the channel and thank you for watching.